Hello and welcome everyone. Today is our second lecture in this series on cellular energetics. And this is probably out of all of them going to be the most challenging. So I really encourage you to take lots of notes. All right, here we go. And again, don't panic. I'm going to do everything in my power to break this down. We'll go from kind of bigger ideas into the details. All right, so what do you need to know? Well, first of all, you need to know the equation of cellular respiration. And we did go over this last time, but we'll go over it again. You're going to want to know at the end of the next lecture, really the differences between fermentation, aerobic, and anaerobic cell respiration. The role of glycolysis, how it breaks apart glucose into two molecules of pyruvic acid, also known as pyruvate. And then, of course, you want to know this process called the prep step that takes the pyruvate from the cytoplasm into the mitochondria and basically introduces it into the Krebs cycle, also known as the citric acid cycle. And then lastly, how chemiosmosis is going to use those electrons from our electron carriers, NADH and FADH2, to produce ATP via oxidative phosphorylation. Now, these are the larger, the bigger ideas. You're pretty much going to need to know everything in the PowerPoint and in the video. So I would go over notes for this unit every single day and do it actively. Don't just look at them and go, oh yeah, I get it. You really want to be active in it. So making note cards, that type of thing. Okay. So part one, this is going to be kind of the basics. So I thought this was kind of funny. When are you planning on finding a job, right? The parent mitochondria says to the younger mitochondria. And don't give me that excuse that you don't have any energy. You're a mitochondria, for nucleus's sake. I don't know. I thought it was funny. Here we go. So what is cell respiration? And we addressed this last time. And basically, it's just when the mitochondria breaks down food molecules. And what food molecules are we referring to here? You said glucose, you're right. And we're going to break them down to produce ATP. Be careful. Do not say that we're producing or making energy, because you can't make energy. You can just transform it or convert it. So there are three stages. Glycolysis. It's anaerobic, it takes place in the cytoplasm, and it produces 2 ATP. The citric acid cycle is aerobic, and it takes place in the mitochondria. And it's going to take place in the liquid portion of the mitochondria, which is the matrix. Lastly, we have oxidative phosphorylation, which takes place at the electron transport chain in the cristae of the mitochondria. And it also requires oxygen. Oxygen is going to be our final electron acceptor here, and it produces the mother load of ATP. So we only get a net production of 2 from glycolysis, 2 from citric acid cycle, and over 30 in this final step. All right, so keep in mind, and we talked about this last time, but I want to make sure that you're clear. The term cell respiration does refer to aerobic, which is using oxygen, and anaerobic respiration, which does not use oxygen. It uses other chemicals. But oftentimes when we just say cellular respiration or cell respiration, we're referring to aerobic respiration. So unless I say otherwise, if I just say, oh, respiration or cell respiration in this video, I'm referring to aerobic respiration. All right, and again, even though we can use lots of different types of carbohydrates, fats, and proteins as fuel, we are only going to be illustrating this with glucose. So this is the formula that you need to know, and you really need to memorize it. It will make your life much easier on the test. Okay, here we go. So these are just kind of an overview of the stages. So 3 stages. Glycolysis, and in glycolysis, it takes place where? If you said cytoplasm, you're right. And it's going to break down our glucose. It's basically going to split it in half. So remember, lyse something is to break it apart. We're going to break glucose in half into 2 molecules of pyruvate. And when that happens, energy is released. And we're going to make 2 ATP. Takes place in the cytoplasm. So the citric acid cycle, also known as the Krebs cycle, is going to fully break down glucose. And this is going to take place in the mitochondria in the matrix. Now, in this particular step, we're going to release a lot of energy and high energy electrons, but those are going to end up being taken away by electron carrier molecules. We're only going to produce 2 ATP here, only 2. And then all of those electron carrier molecules from glycolysis in the citric acid cycle are going to end up at the electron transport chain, where through oxidative phosphorylation, we'll go through what that means, and a process called chemiosmosis, this is where we're going to make most of the ATP. Okay, so this is kind of just showing an overall picture, and I am so sorry about that. I dropped a dog toy on the floor. It's just showing an overview. So again, the first process is glycolysis. Glyco for sugar, lysis to break apart. We're breaking apart a 6-carbon glucose molecule into 2 3-carbon pyruvate molecules. All the high energy electrons from breaking this apart are going to be taken by an electron carrier molecule called NAD+. Once it accepts those electrons, what element is also going to attach to NAD in addition to the electrons? And by looking at this, hopefully you determine that it was hydrogen, because that's where our electrons are going to, what our electrons are going to move with. These are going to go to the electron transport chain. So those are gone. But we do produce 2 ATP net. And this is through substrate level phosphorylation, and we'll talk about that. But basically, we're just going to, use an enzyme. We're going to attach a phosphate, and we're going to produce ATP. Next, one thing that's not labeled in here is the prep step. And I would label that, especially if you sketch this out in your notes, which I would, label this prep step. And so we're going to convert this pyruvate into acetyl-CoA. That's going to go into the citric acid cycle. Now, we're going to complete the breakdown of glucose by completing the breakdown of pyruvate, which has been converted into acetyl-CoA. And again, don't panic if you're like, oh, so many details. We're going to go over all of this. This is just overview. And we're going to produce 2 more ATP via substrate level phosphorylation. We are going to be releasing a lot of high energy electrons, and those are going to be taken by NAD, which again is an electron carrier molecule. Once it's been reduced and it's got the high energy electrons, it becomes NADH. It goes to electron transport chain. Now, we also have another electron carrier molecule that's only seen in the citric acid cycle. It's not seen in glycolysis, and that's FAD. And that's going to take high energy electrons and the hydrogens and become FADH2. And these basically have a very similar function. They're taking high energy electrons, which we're going to use with chemiosmosis to produce lots of ATP through oxidative phosphorylation. And again, this occurs at the electron transport chain in the cristae, that's those folded parts of the mitochondrion. Okay. All right, so the structure, because I've been mentioning matrix and I've been mentioning cristae. So this is a mitochondrion, and mitochondrion is singular for mitochondria, which implies many mitochondria. Many of us, including myself, will sometimes refer to a singular mitochondrion as a mitochondria, and that's technically incorrect, but it's what a lot of us say. So I hope this isn't you. All I learned from biology is mitochondria is a powerhouse of the cell. Now you're going to be able to know why it's the powerhouse when we finish with this. So we have the matrix. That's going to be this liquid portion of the mitochondria, and this is where the citric acid cycle, also known as the Krebs cycle, takes place. And we don't produce a ton of ATP here. In the cristae of the mitochondria, this is going to be the location of the electron transport chain, where we do oxidative phosphorylation and chemiosmosis, and this is where we produce the mother load of ATP. All right, here we go. And you should know the structure of all mitochondria. Okay. So check for understanding. I know this is earlier than usual, but I'm going to break this down into a few checks for understanding just to make sure that you're not totally lost. So this stuff you're going to need to know going forward. So the first is, what is the chemical equation for cellular respiration? And then in the conversion of glucose and oxygen to carbon dioxide and water, which molecule is oxidized? And yes, I know we went over this before, but I really want to make sure that you're clear. And then which molecule is reduced? All right, what happens to the energy that is released in this redox reaction? So where do those high energy electrons go? NAD plus is called A, and its reduced form is, all right, here we go. So now we're going to get to step one, or stage one. This is glycolysis. So here we go. So glycolysis, again, occurs in the cytoplasm or the cytosol. So you might look at this diagram and it makes you want to barf a little bit because it's like, I don't even know what's going on here. Don't panic. It's all good. So this is showing glucose. And really what we're concerning ourselves with here are the carbons, just to kind of figure out, okay, where are these carbons? You know, what are they being turned into, et cetera. All right, so glucose is going to ultimately get broken down through glycolysis, into 2-pyruvic acid. Okay, so we have to use 2 ATP to start this reaction, right? That's the activation energy. So we call this the energy investment phase. And again, we use 2 ATP, and we're going to phosphorylate. So 2 ATP to break this apart. And we phosphorylate one from each ATP, these 3 carbon molecules. Are you with me? And these are called, this is a temporary intermediary called PGAL. And you're going to see this later when we get into photosynthesis. Okay, now, what's going to happen next is that we are going to produce ATP. Okay, now the ATP, we're going to have another intermediary that's not even named here. And it's going to be phosphorylated again. Okay, so we're going to end up with 4 phosphates. So you're going to have one here and one here. So it's going to be a biphosphate on each molecule. And ultimately, those will be removed. We're going to make 4 ATP. Now, you might say, okay, you told me earlier that we make 2 ATP and now it's saying 4. You must be wrong. Well, it's 2 net because we have to, before we say how much we have to pay back the 2 that we used. So you can kind of think of it like I borrowed $2 from you and I made $4 off of it. So now I have to give you your 2 back. So I've only come ahead by 2. So we come ahead with 2 ATP. That's the net. It's kind of like a net paycheck, right? Your gross paycheck is what you earn, and then you have to pay your taxes. So we're paying our taxes. We're paying back the 2 ATP. All right, next. You'll see here that we have 2 NAD plus that are going to be released. Now, where does that come from? That is, we have these coenzyme electron carrier molecules. So NAD is an electron carrier molecule. And when it accepts those high energy electrons, it's also going to be accepting hydrogen. So remember, when we do these redox reactions, and this is a redox reaction, the electrons travel with hydrogen. So it forms NADH. And this is going to go to the electron transport chain, and we'll use these electrons later. Now, the 2-pyruvic acids are going to go into ultimately the Krebs cycle, but we have to do the prep step first, but let's not get ahead of ourselves. Okay. So glycolysis, I've said this before, and I know that I'm a little repetitive with this, but I really want to hammer this stuff home. So if you're like, all right, I know it. Good, I've done my job. So glycolysis is sugar splitting. Glyco for sugar, lysis for break apart. And they believe that of all these processes, this one is the oldest. It is anaerobic. So if we think back to early prokaryotic cells, early bacterial cells, photosynthetic bacteria like blue-green algae, there was no oxygen available. So they were still able to carry this process on. Additionally, it does occur in prokaryotes. There is no mitochondria. If you remember endosymbiosis theory, it's believed that mitochondria formed from these early prokaryotes that had a membrane inside, like a cristae. So this is why it's believed to be a very old process. They call it a metabolic heirloom. Again, it occurs in the cytoplasm, and it partially breaks down glucose into 2-pyruvic acid, which are 3 carbon molecules. So it's basically splitting that 6 carbon glucose in half. All right. The other thing to keep in mind, and you want to memorize these net products, 2 ATP, and then two NADH. And that NADH goes where? If you said it goes into the electron transport chain, you're totally right. All right, here we go. Oh, additionally, water is produced here. So if we look at our formula, we're going to see a production of water. Okay, here we go. And I already mentioned that oxygen wasn't required. All right, so step one, the energy investment stage. This is where we use ATP. How much ATP is used? If you said 2, very good. So we're basically going to use this is the activation energy, to break apart that glucose and phosphorylate them. And that's going to make 2 P-gals, which are temporary. Next, stage 2, this is the energy payoff stage. This is where we're going to make 4 ATP with a net production of 2 ATP. Okay. And so it says that these 2 3-carbon compounds are oxidized. That's the P-gal. And so we're going to end up making pyruvic acid, also known as pyruvate. Okay. So 2 net ATP produced, substrate level phosphorylation, which we'll get into. And then we also have 2 molecules of NAD plus, and I don't know why it made a little question mark there, but it did, into 2 molecules of NADH, and those go where? All right, electron transport chain. Very good. Okay, so we're briefly just going to look at the structure of NAD plus. What does NAD stand for? It stands for nicotinamide, but you just need to worry about NAD plus. So this is NAD plus. This is the oxidized form. The reduced form is when it accepts those high energy electrons from glucose. So this is an enzyme that's used. It's called dehydrogenase. And it basically is going to help to reduce that NAD plus into NADH. And one really cool thing about NAD is it's reusable. So once it releases those high energy electrons, which is going to happen later at the electron transport chain, that NAD can be reused. If the NAD could not be reused, all of this would stop. And this is really important to keep in the back of your head, because when we get into fermentation, this becomes a real problem for the cell. All right, here we go. So what is the substrate level phosphorylation that I've been talking about? Well, basically, we have an enzyme here, and here's our substrate, right? So our substrate would be basically ADP. And so what's going to happen is we want to add this phosphate to ADP. And the energy is going to be used from our reaction, from glycolysis, to do that, right? And so we make our ATP. And this is going to be a temporary molecule here, this substrate, which you don't need to worry about. If you want the details, I'm happy for you to come look at my biochem book, but don't worry about this temporary substrate. Okay, what we're doing here is just generating the ATP. Okay, so this is our substrate. I guess I can get into it a little bit. So here we would have P-gal, for example. So we would have P-gal here. We would take the phosphate off and attach it to ADP. This is going to give us pyruvate and ATP. So, but again, I wouldn't stress too much about all of these mechanics, but if you're curious, okay? All right, here we go. Now, when we start looking at substrate level phosphorylation in the Krebs cycle, we're not using P-gal, which is why I don't want you to memorize all this detail. You will get to do that when you take biochem in college. Okay, so again, we're doing some intermediary checks for understanding. So number 1, why is glycolysis considered an ancient metabolic process? Where in the cell does it occur? Does glycolysis occur? What are the reactants and products of glycolysis? Okay, which has more energy available? And keep in mind, as you break something down, you're releasing energy. So what has more energy, ADP or ATP? What about NAD plus or NADH? And now we haven't talked about FAD in any detail, but it's an electron carrier molecule like NAD. So what do you think? FAD plus or FADH2? All right, where does glycolysis? Oh, I already said where does glycolysis take place? Ignore that, and ignore the main products. But because we already mentioned those, I want to know where all those products go. Okay, here we go. All right, this is a video that I want you guys to watch before you move on. I'm not going to assign an ed puzzle. I'm going to trust that you watch it. This is Savage, and again, his voice is very mellow. But his diagrams are excellent. And I think hearing it again from somebody else is going to help you with this. So I really, really, really want you to watch this. And I'll put the link in the ed puzzle. Okay. Now I would take a little break and then we're going to get started with the Krebs cycle. So the Krebs cycle, as it says in our meme, Krebs cycle occurs in the Matrix. So that's Keanu Reeves, right, from Bill and Ted's Excellent Adventures when that picture is taken. And then, of course, he was in the Matrix. This is a different Matrix, the Matrix of the mitochondria. But anyway, so we're going to look at stage 2 of cell respiration, which is first the oxidation of pyruvate, which is the prep step, and then the citric acid cycle. So this is the prep step. And again, it looks super confusing and, people are like, what is this? It's not bad. So here we have pyruvic acid, also known as pyruvate. And I'm sorry that there are multiple names for things. If I were in charge of the world, there wouldn't be multiple names, but there are. And I don't know what they're going to use on the AP exam, so you want to know these. So we've got our pyruvic acid. Where was this pyruvic acid produced? So what process and what location? If you said glycolysis in the cytoplasm, you're right. And so it's going to pass through the mitochondrial membrane, okay? And then one of these carbons is going to be broken off, okay? And when that happens, right, we're releasing high energy electrons, which are going to be picked up by NAD to make NADH. And where does this go? If you said it goes to the electron transport chain, you are correct. So once this byproduct, and we're not even going to name this one because I don't want you getting bogged down in that. This byproduct here is going to be added to a coenzyme A. Okay, so coenzymes are wonderful little things, and this coenzyme bonds with this to make acetyl-CoA. What does CoA stand for? It stands for coenzyme A. Okay, so if oxygen's present, we're going to have the citric acid cycle take place. And you might say, well, wait, where does this oxygen come from? From this carbon? Well, you want to think about the fact that there's oxygen in this molecule. It's not oxygen gas, but there is oxygen present in pyruvic acid, and that's where it comes from. Okay. Now, so we've converted it to acetyl-CoA. Now, acetyl-CoA is going to go into the matrix where we're going to do the Krebs cycle. So here we go. And this is one of those things in the book. It looks so confusing. And if you start looking at some videos online, you're going to see 50 million steps. There's going to be like redox reaction after redox reaction and all these intermediaries, malate, succinate, etc. You do not need to know those. You need to know that they occur, but you don't need to memorize all that detail. You'll do that again in biochemistry. Okay, so this is just showing a nice little diagram of pyruvate oxidation. So remember how I told you there were some oxygens here? This is going to be the part that gets removed with the CO2. Okay, so this is pyruvate. Okay, and so what's going to happen here, and this came from originally a carboxyl group, but the hydrogen is gone. Okay, but this was originally a carboxyl. So now we're going to remove this to produce CO2. Again, no atmospheric oxygens necessary. When we break this off, right, this hydrogen and the high energy electrons, when this happens, are going to go to NAD, okay, to make NADH. And again, this is going to go to the electron transport chain in the cristae. All right. Coenzyme A will then be added to this molecule here, which is acetyl-CoA. And this acetyl-CoA is really, really important because we're going to use this to make citrate, which is the name for citric acid. So this becomes very important. Without this input here, we can't do the citric acid cycle. So, all right. So CO2 is produced. It is completely and totally a waste product. This is why we make CO2 when we do respiration. And also NADH is produced. Okay, here we go. So this is showing a very general picture, and we'll get into some more details, a little more detail. So again, you might look at this and go, this is horrible. It's not that horrible. And my piece of advice would be even to screen capture this, and then you can annotate it. So here's our pyruvate. What process created pyruvate? If you said glycolysis, you're right. And next, we oxidize this, right? So we are going to remove the high energy electrons from this. We're also removing a carbon to produce CO2. We add coenzyme A to make acetyl-CoA, which is going to enter the citric acid cycle. Now, where does this take place? If you said inside the mitochondria, you're correct, and this is the prep step. So I would label this prep step. I would label this glycolysis if you print it out. So what's going to happen here is acetyl-CoA is going to come in, and it's going to combine with a 4-carbon molecule to make citric acid. And then really, the rest of this is breaking down citric acid again, and then regenerating that 4-carbon molecule, which is called oxaloacetate. And we'll look at that in a second. When we're breaking this back down, the citric acid, we're releasing more carbon dioxide. And we're releasing, as we are breaking down the rest of this glucose, because it was originally glucose, the acetyl-CoA and these parts that are in citric acid originally came from glucose. We are going to be releasing more high energy electrons that we're going to need at the electron transport chain. So NAD plus takes those high energy electrons and goes to the electron transport chain. Where is the electron transport chain located? Be specific. If you said in the cristae of the mitochondria, A plus, you get a cookie. Okay. And then we're also generating ATP here. Does anybody remember how much ATP from when I said earlier? 2 ATP. All right. And then we're also, as we continue regenerating this oxaloacetate that we're going to use for the next acetyl-CoA that comes in, we are going to be releasing more high energy electrons that go to where? If you said electron transport chain, you're right. And then we're going to regenerate that 4-carbon molecule, and this is going to happen again and again. And this is, it's a cycle. We could really start anywhere with this. You do want to kind of be able to explain these processes. And if you're confused, I would watch it again. All right. So another name for the citric acid cycle is the Krebs cycle, and these are used interchangeably. Krebs is the guy that discovered these processes. So again, just a summary. The citric acid cycle, or the Krebs cycle, it occurs in the matrix of the mitochondria. That's a liquid portion. And remember, we have to have the prep step first to create the acetyl-CoA. And the acetyl-CoA, again, I don't know why these question marks are here. We'll just have to ignore them. But acetyl-CoA is going to attach to this molecule, this 4-carbon molecule, called oxaloacetate to form a 6-carbon citric acid, from which this is named. Another name for citric acid is citrate. Did you notice pyruvic acid is also called pyruvate? So you can get an idea that this ATE oftentimes is going to refer to an acid. And remember, CO2 is going to be reduced when we start breaking this back down again. Ultimately, and you should memorize this, we are going to gain a total of 2 ATP. But you might say, why is this even important? It's such a puny amount. Well, we have totally broken down our glucose, because remember, glycolysis only really breaks it down in half. Here, we've really broken it down. So we have 6 NADH and 2 FADH2, which are bringing those high energy electrons from glucose to the electron transport chain. The ATP is also produced by substrate level phosphorylation. So not a lot produced. Now, here's a little bit more detail. And again, this might make you want to barf, but it's really not that bad. So we're going to just kind of step through it. All right. We're going to start here with oxaloacetate. Oxaloacetate is another one that is oxaloacetic acid or oxaloacetate. So this is a 4-carbon molecule. When the acetyl-CoA is introduced, it combines with that oxaloacetate and it forms citric acid. Okay? Now, this is temporary. What's going to end up happening here is we're going to start breaking it down. So we're going to break off 1 carbon first. We're releasing high energy electrons. So we're going to form some NADH here, and we're releasing CO2. And then this is going to happen a second time, right? And we're forming more. And now we have a 4-carbon molecule. We still are going to break some high energy electrons off of this. And we're not showing you all those details. You do not need to know that. Okay, but basically what we're going to do here in these steps where we form the FADH2 and where we form the NADH, what we're doing here is recycling oxaloacetic acid. So to make the oxaloacetic acid, we're taking more high energy electrons off. And again, those are going to go to the carriers. Now you might say, wait a second. These numbers don't add up. You said 6 NADH and 2 FADH2. I'm only seeing 1, 2, 3 NADH and 1 FADH2. So, Robson, you were wrong. Now, it may be the case that I'm wrong about things because as much as I like to think I'm perfect, I really don't think I'm perfect. And I make mistakes just like everybody else. But keep in mind, when we have a glucose, 1 glucose, we break it in half and we produce how many pyruvic acid? If you said 2, you're right. And then each one of those pyruvic acids gets converted into acetyl-CoA. So for 1 glucose molecule, we're going to do this cycle 2 times. Okay? So 2 acetyl-CoAs will go in. So we just double this. Okay? If you have questions, let me know. Here's our summary, our inputs. So from 2-pyruvate, which come originally from glucose, right? We get these from what process provides the pyruvate? If you said glycolysis, you're right. Very good. And then when did we generate this acetyl-CoA? If you said prep step, you're right. And then this combines with the oxaloacetate or oxaloacetic acid. And then it produces the citric acid, which we break down, right? And then we regenerate this. And ultimately, we've got the 2 FADH2, 2 ATP, 6 CO2. Now you might be like, wait a second, Robson. I thought you said there were 6 NADH, and I understood why we got 6. Where are the other 2 from? Those other 2 come from the prep step. So they're kind of combined together in this. So that's where the other 2 come from. You're not crazy. It's 6 in the actual cycle itself and 2 right before. Okay, so again, the Krebs cycle or citric acid cycle. It's got 8 steps. You do not need to memorize all the details of those steps. Just know that each one has its own enzyme. You don't need to memorize those. You can do that in biochem. Fun times lying ahead. And it's a lot of redox reactions. And keep in mind, again, that acetyl-CoA from our prep step, which originated as pyruvic acid, joins by combining with the oxaloacetate, and it forms citric acid or citrate. Right? And then the next 7 steps are just kind of doing redox and converting that citrate back into oxaloacetate and taking all the high energy electrons out. Oxaloacetate is a low energy molecule because we've taken the high energy electrons. All right, now this is a really important part, and I would circle this if I could. The NADH and FADH2 that are produced by the Krebs cycle are going to take all those high energy electrons that have been extracted to the electron transport chain. Now it says here, extracted from food. What food are we using to exemplify this? If you said glucose, you're right. All right, here we go. So this is showing the entire cycle. You do not need to memorize all of these details. You'll have to at some point if you're going to go on in biology, biochem. You will have to memorize all these. You do not need to do it now. That's later. Okay. You'll notice here we have this molecule GTP. Probably like, what is that? We'll see GTP more later, but GTP works just like ATP, except it's guanosine triphosphate. So it uses a guanine instead of an adenine. All right, here we go. You should know oxaloacetate, not the structure, as a 4-carbon molecule. And you should know citric acid is a 6-carbon molecule that's formed by combining oxaloacetate with acetic acid. But you don't need to memorize the rest of these intermediaries. There's no reason to. All right, again, Craig Savage has another video, and I know his voice is really soothing, but seriously, it's awesome, and it's a fantastic review. So I would definitely watch this again, especially before we proceed. And you should absolutely take a break before we get into this next part, which is going to be the final part of this video. All right. Here are the checks for understanding. So what happens in the prep step or prep phase before the Krebs cycle? Where does the citric acid occur in the cell? And then specifically, where in its location does it occur? And then what are the main products of the citric acid cycle, which I just abbreviated there? Where do those main products go? Okay, so make sure you've taken that break. Take it now. All right, I hope you've taken a break because now we're going to get into chemiosmosis. This should be an S here. The formatting got a little messed up. Oxidative phosphorylation, which takes place at the electron transport chain. Be careful. The electron transport chain is not a process. It is not a process. Do not say that it is a process, either on my test or the AP exam, because it's not. The processes that occur, really, the process is oxidative phosphorylation, which is driven by chemiosmosis, and we'll talk about that. And I thought that this was hilarious because there's so many people who like to, use as big of words as possible, and I thought this was funny. I heard you like to phosphorylate your glycogen phosphorylase. So I brought you some phosphorylated phosphorylase kinase so you can phosphorylate your phosphorylase while you dephosphorylate your phosphorylated glycogen. And it sounds like impossible, but if you understand phosphorylation, it should make a little bit of sense. Okay. All right. So oxidative phosphorylation is different, very different than substrate level phosphorylation. And pardon me while I take a quick sip here. I've been talking for a while. Instead of using a substrate like PGAL, we instead are going to be using chemiosmosis to drive this. And basically what happens is chemiosmosis is going to work with this electron transport so that they go through an enzyme and a channel called ATP synthase to drive ATP synthesis. So that may just sound like absolute nonsense right now, but it'll make sense in a minute. So again, after glycolysis in the citric acid cycle, we have all these high energy electrons that are going to be in these electron carrier molecules, NADH and FADH2. And it says, that these high energy electrons, right, account for most of the energy extracted from food. What food? Glucose. All right. So these two electron carriers are going to take these high energy electrons and bring them to the electron transport chain. The way they work is really, really similar. The only real difference is FAD will bring them in a little bit later in the chain. But honestly, very similar. So typically you're going to see this illustrated using NADH. Okay. So oxidative phosphorylation. So what do you think with this title, oxidative phosphorylation? What do you think is ultimately going to be driving this other than chemiosmosis? The hint is oxidative. If you said oxygen, you're right. So let's take a look at this. So electron transport chain, where this occurs is in the cristae of the mitochondria. And the cristae is the inner membrane. Now, it'll produce somewhere between 26 and really it can even be as high as 34 ATP, so a lot more than 2. Okay, so what is chemiosmosis? What's going to happen is after those electron carrier molecules come and drop off their electrons, the energy in those electrons are going to be pumping these hydrogen ions. And you might say, where did hydrogen ions come from? Well, remember, they are taken along with the electrons. So when NAD drops off the electrons, it's also dropping off those hydrogens. And the energy from the electrons pushes those through that mitochondrial membrane, right, in the cristae. And what's going to end up happening is you're going to get this super high concentration of hydrogens up here. And they're going to go through an enzyme, which also acts like a channel. It's like super cool. And this is called ATP synthase. So you may have noticed that catabolic enzymes are often named after what they break down, right? So lactase is named after lactose, right? And it breaks it down. Well, anabolic enzymes that build things, tend to be named after what they build, but you'll often see synthase after it. So in this case, it's ATP synthase because it synthesizes ATP. All right. So hopefully that makes a little bit of sense. All right. Again, and I know I'm repeating myself. Hopefully you're like, oh, I know this already. I'm sick of hearing it. But if you're not there yet, then it's for you that I'm repeating myself because this stuff is tricky and there's a lot of detail. So again, the electron transport chain, which sometimes we abbreviate ETC, is located in that inner membrane of the mitochondria, and that's called cristae. Now, when we look at the chain, and we're going to look at a diagram in a little bit, most of these are proteins, okay? And some of these have cytochromes in them and, other things, and these are like protein complexes. And what's going to end up happening when the electrons kind of move down this chain, they, will become reduced when they receive it. Then they give it up to the next one and they become oxidized, et cetera, et cetera, et cetera. And as they go down this chain, through all these redox reactions, free energy is going to drop every single time. And then at the very end of the chain, they're going to be very low energy, right? And finally, oxygen is going to be the final electron acceptor. It's going to accept the electron that's coming down the chain and the hydrogens that are going to go through ATP synthase, and it's going to form water, which is very, very low energy, right? Because we've already taken all that energy out. Okay. Now, how do those electrons get there in the first place? NADH and FADH2 from glycolysis and the Krebs cycle. So again, it's a lot of protein complexes. And I mentioned cytochromes. Why are those important? Well, because they have iron atoms. And if you think back to hemoglobin, hemoglobin has iron because it attracts oxygen. Well, they have the same kind of function there, here too, right? That cytochromes are these proteins with iron atoms, and they attract oxygen because we need oxygen for this process to work. If we have no oxygen, you already know if people get deprived of oxygen, they drop dead. Why do they drop dead? Because they can't do cell respiration. Okay, the electron transport chain in and of itself does not generate ATP. It's not a process. Okay, it is not a process in and of itself, and it does not generate the ATP. Okay, its job basically is to, kind of allow this energy to be released from food. From what food? Glucose, right? All that energy from those electrons is going to be released as it goes through these, the series of redox reactions down the chain. So it's releasing in manageable steps. We're going to take that energy to push those hydrogens across, and we're going to use that to drive ATP synthesis. It's such a cool process, and I know I get very excited over lots of things, but this is like a super cool process, and it's amazing. It's just, to me, amazing how energy efficient it is and how awesome it is. Okay, so the electron transport chain, again, it's a bunch of molecules that are going to be embedded here in this inner chain in the cristae, and these are typically protein complexes, right? There are a whole bunch of them. So NADH brings high energy electrons through here, right? It's going to drop them off with their hydrogens, then NAD plus is released so that it can go back to glycolysis and keep doing its thing. Same thing with FADH2, right? It just drops them off a little bit later here. Now you might say, all right, what is this? CYT, that's cytochrome, right? This is showing iron, right? So these are different cytochromes here. Okay, so basically the key is here when it drops them off, right, this first part will become reduced, then it gives it up, this is oxidized, now this is reduced, and it just continues going down here. But the ATP is not made here. This is really important. Okay, it just allows little bits of energy to be released each time instead of this huge amount that we wouldn't be able to really deal with. Okay, now ultimately at the end, these are now low energy electrons. They're high energy here, low energy here, right? And these electrons that are coming out were originally from our carriers and ultimately were originally from glucose. And this is going to combine, these will combine with hydrogen, and we'll talk about where that hydrogen's coming from, with oxygen to make water. Okay. All right. And these arrows should be yield, but all right. Take a deep breath. Okay, that's a lot of information. So what's going to happen is this electron transfer, we said it's releasing energy. And what that energy is going to be used for really is to power our pump, to pump hydrogen ions against the concentration gradient, right? So they're going to be going up here. There's going to be a high concentration, if this is our membrane and NAD is coming in here, it's going to be releasing the hydrogens and the oxygens at the beginning of the chain. So here's our chain going down. And so the electrons will start going down here. When the energy is released, it's going to pump hydrogens through. Low concentration, high concentration. So we're going against the concentration gradient. And we're going to end up with a whole bunch of hydrogen here. Okay? Now, we're going to have ATP synthase. And you see here, it calls it a channel. You might be like, wait a second. Robson, you just said it was an enzyme. Well, it is an enzyme, hence why it ends in ACE, but it's also a channel and it's really cool. So what's going to happen is we have this buildup of hydrogen, and the hydrogen cannot diffuse back through that membrane, right? Because it's going to have that nonpolar inside. So it's going to be stuck up there. And the only place it can go is through ATP synthase, right? So it starts going down there. And ATP synthase is going to use this flow of hydrogen ions, and there's going to be, exergonic flow of hydrogen. So energy is being released, and then ATP synthase starts spinning. It's like super cool, and that drives the phosphorylation of ATP. This is chemiosmosis. So it's basically using the energy in a hydrogen gradient, which is also called a proton gradient. This is also called a proton pump. You might be like, why is it a proton pump? Because a hydrogen ion, H+, is nothing more than a proton. So it uses this energy in this proton gradient or hydrogen ion gradient to drive cellular work. That is chemiosmosis. And chemiosmosis is required to do oxidative phosphorylation. So here's another picture. So, and this is less complex and I like it better. So NADH is going to drop off those high energy electrons and hydrogens, right? So, and it becomes NAD plus, and it's going to go back, either into the matrix or into the cytoplasm to be reused. So high energy electrons are going to be, going down this chain, doing lots of redox reactions, right? And these are also, these proteins are electron carrier proteins, and they pass them along. Okay. Now, when this happens, energy is released and it's going to start pumping through hydrogens. Okay. And FAD is going to enter a little bit later on, but it's basically the same process. So now we're going to get all these hydrogens, and it's a lot more than this. You definitely want to look at Mr. Savage, Craig Savage's video on this, because I think the way that he kind of animates it all, although it's not totally animated, but it's much cooler than what I have here, and I think it explains it better. So we're going to end up with this huge concentration of protons or hydrogen ions up here, and they're going to start going through this enzyme. So it just says enzyme. What is the name of this enzyme? If you said ATP synthase, good for you. It's also a channel. So these are going to start going down here. This is going to start to spin, right? And that energy is going to drive this phosphorylation, which is oxidative phosphorylation. So you might say, well, what's the point of the oxygen here? Well, the oxygen basically is going to take the hydrogens from here, as well as the low energy electrons that are released at the end of the chain, and take them away. If oxygen disappears, then hydrogens get stuck. They get clogged. So kind of think of, what happens if we're at the gate of the Air Force Academy, right? And all of a sudden, car doesn't go through, maybe they're searching student backpacks or, trunks and that kind of thing. And all of a sudden, that flow of cars isn't working and it backs up and movement stops. It's the same kind of idea. If oxygen's gone, hydrogen doesn't move through. And so everything stops. But as long as we've got oxygen, it's going to take this hydrogen, and it's also the final electron acceptor. So these proteins are accepting electrons until the last one releases it, and it is going to be accepted by the oxygen. So the role of oxygen in cell respiration is the final electron acceptor. And again, if that oxygen is not here, everything stops and you die pretty fast. Okay, so this again is just showing what would happen if we didn't have an electron transport chain, right? You'd have this huge release of energy without, and in this case, there's light energy here, right? Heat energy, and your cells wouldn't survive it. So the reason why the electron transport chain is so useful is because it's a controlled release. So we're releasing just in tiny amounts. All right. So again, just to kind of go through this again, the energy that's stored in this gradient, right, where we have all these hydrogen ions, all these protons, right? This energy that's stored up there couples the redox reactions of the electrons here to really drive that oxidative phosphorylation. Now, this hydrogen gradient, we call this proton motive force, right? Because those hydrogen ions are protons. So basically, motive meaning movement, this force is going to cause movement of ATP synthase, right? Which is a lot of energy that then can be taken to phosphorylate that ATP. So it's pretty cool. I thought this was funny. The inner mitochondrial Doge meme. I don't know. It made me laugh. You might think it's funny. Maybe you think I'm just such a nerd. I am a nerd. But I still thought it was funny. Much electron transfer. So cytochrome, oxygen consume. Wow. Yeah, epic. Okay, so this is another picture basically showing the same thing, right? Electrons move through the electron transport chain, and then we have proton pumps, right, that are going to be moving this hydrogen across using energy that's released when this happens. And ultimately, where are all these protons going to go? If you said they're going to go to ATP synthase, you're right. What then happens to ATP synthase? If you said it spins, you're right. And that's going to drive ATP synthesis by what? If you said oxidative phosphorylation, you got it. Okay. So again, chemiosmosis works with ATP synthase. It works with the redox reactions in the electron transport chain. So this is showing the picture of, and it's really cool. I mean, it looks like something that engineers would have designed. And I'm telling you, you engineer type people who are taking this course, a lot of engineering is modeled off of biology. And I mean, if you look at this ATP synthase, it has a rotor, right? It's got a rod. It's just like super, super cool. Anyway, this will spin like crazy, which will allow for that phosphorylation there. And it's super cool. I don't know. I hope you think it's as cool as I do. Okay, and here's another picture again. I would, if I were you, one way that I would try to study for this would be to look at this picture and try to explain each step and what's going on. And if you get stuck, let me know. Okay, here is the Savage video. And again, I find it so funny his name is Savage because, you're like the Savage video. And it sounds like it's going to be so savage. And then he's like, today we're going to talk about, but seriously, he's awesome. Definitely watch this. Please, I'm begging you. Okay. So just a little bit of an accounting of ATP production. So when we're doing cellular respiration as a whole, we go right from glucose in glycolysis. We're going to start breaking it apart. We break it apart in glycolysis and finalize the breakdown in the Krebs cycle. NADH and FADH, FADH2 are going to take all of these high energy electrons to the ETC, where we're going to generate ATP via oxidative phosphorylation, which happens because of chemiosmosis, which is this proton motive force. Now, 40% of the energy in a glucose molecule is going to be transferred to ATP. And that's pretty energy efficient, believe it or not. And it makes anywhere, from 20 something to 38 ATP. It can make a lot of ATP, right? It's crazy. Now you might say, wait a second, Robson, you said less than that earlier. Well, think about, we've also got 4 ATP that we made, first 2 from glycolysis, 2 from the Krebs cycle, and then, maximum of like 34 during oxidative phosphorylation. So this is showing an overall of this entire process. And this is something else that I would go through when you're studying and try to explain it step by step. And exactly how each part happens, what the products are, where they go, the locations, that type of thing. Okay, checks for understanding. And this was made, this made me laugh. This was made by a student a couple years ago in AP biology. They made this meme. So here are the electrons that are in NADH and FADH2, right? Those are the electrons. Here are the hydrogen protons. And then these guys are like, oh, hey, electron carrier proteins of the electron transport chain. I don't know. It makes me laugh. Anyway, all right, here we go. Checks for understanding. Where does the energy come from for chemiosmosis slash oxidative phosphorylation to generate such a large amount of ATP? How is a proton gradient generated? What's its purpose? Describe how ATP synthase works. And then in cell respiration, how many ATP are generated through substrate level phosphorylation? And how many ATP are generated through oxidative phosphorylation? I really hope that that was helpful. You probably have a lot of questions, so please don't hesitate to ask them here in the Ed Puzzle. And again, like I've said before, if you're at a point where you're like, I don't even know what questions I have at this point, I'm confused, please rewatch this video. I would definitely recommend watching the Savage videos that I've posted. You can watch Bozeman additionally, but watch Savage first. There are a lot of videos out there that you can watch that will help, but do not hesitate to email me questions as well. And I hope that this was helpful. Please let me know. And as always, please feel free to leave suggestions for how I can make these better. And thank you for staying with and watching this a long time.